Let's see. Okay, so today we're going to keep talking about sequence data, and we'll start talking a little bit about how to align sequences to each other. And eventually I'll show you how to do a statistical assessment of the quality of an alignment. So to start, I wanted to we're going to go back to the seqanalysis.r script, and let's see where we left off here. So we talked last time about the seqnr package as a way to get sequence information for a gene or a protein. And we talked about the fact that when we do this, we get multiple versions, multiple sequences often for the same gene. We talked about the fact that one gene can be spliced together in different ways to give us different proteins with different functions. So that's what we're getting out. We're getting different sequences for subsequences, basically, of the whole genomic location that contains the gene we're looking at, that are known to result in specific protein products. Introduced you to FASTA files as a format for representing sequence data. It's just a text file, and it has a specific way that you put the sequence information in there. In addition to the actual sequence, the ACTG sequence for a gene, there will be potentially some additional information, such as a quality score for each base in the sequence. So if we ask for the sequence information for the Mike gene, depending on where we get it from, there is the possibility that there would be an entry in the FASTA file for sequence 1, A, T, A, C, G, ... And for each one of those letters, a different code. I don't think it's in this FASTA file example, but we'll see it in another one. A different code with a letter, it corresponds to each of the letters in the sequence, and that codes for confidence or a probability of a correct match, something like that. So often when we have these sequence data information, we have the sequence, but we also have some measure of how good of a sequencing effort it was. Are we confident that this is the actual sequence, or were these all just coin flips? I couldn't tell if it was an A, so I picked an A or a T at chance. All right, we left off here, and I was going to talk briefly about what you can do with sequence data. One thing that you can do with sequence data is to characterize its kind of global or large-scale characteristics. Something as simple as, what are the proportions of A's, C's, T's, and G's in the sequence? Suppose we have a gene that, like in the homework 2, let me pull that up real quick. We already have it. So like in the homework, this gene you're going to consider H1F0. This is a gene, same gene name, but it's present in multiple organisms. So you have it, a mouse has it, and a cow has it, for example. So it would be interesting maybe to compare the H1F0 gene sequence in humans to that in mice and to that in cows. One way to compare it is to say how many matches and mismatches are there, because our sequence is probably not going to be the exact same as the mouse sequence or the cow sequence. We could say, what are the proportions of A's, C's, T's, and G's in humans? What's the proportions in mice? What's the proportions in cows? That would be one way to say how alike are these two sequences. Back here in our script file, I mentioned this idea of GC content, which is kind of another high-level way to characterize the content of a sequence. And all GC content refers to is what proportion of the bases in the sequence are either G's or C's. You could have A's, T's, C's, or G's. Some sequences will have lots of G's and C's. Others will not have as many, just depending on, how it is made up. Why care about G and C? What's interesting about GC content? I'll just briefly show you that there's a Wikipedia article just about GC content. And if you read through here, it talks about genes that have a lot of GC, high GC content, are a little bit different structurally and in terms of their characteristics than other genes that have less GC content. In particular, so here's the letters A, T, C, and G. In the double helix of DNA, A's want to bind to T's. They are set up physically so that they line up well and stick together. C's and G's similarly like to go together. And one thing that's different about how an A and a T goes together versus a C and a G goes together, there's two places, there's two bonds, there's two chemical bonds that connect A and T, whereas there's three that connect C and G. So if you were to just think about sticking magnets together, if you had two things and you stuck them together with two magnets versus two things and you stuck them together with three magnets, there's going to be more stability. For example, if in the presence of high temperatures, a high temperature can break these bonds, which could result in problems with the DNA. Things pull apart, you get mutations. C and G connections are more stable. And so it can result that, let me find a good summary sentence here. So we'll just say this. Sequences with high GC content, DNA with lots of GC content is more stable than DNA with low GC content. So structurally, high level, how does this gene in humans compare to that gene in mice? We could talk about what proportions of the different letters there are. We could talk about the GC content. Humans have 62% GC content. I don't know, I'm making that up. And cows have 48. Okay, so that's just something that shows up in the literature. If you're reading papers about some of these applications, you might see people talking about GC content. That's all it is. Let's run a little bit of code here. I'm going to get us seq_nr libraried in. I mentioned this last time, but I'll reiterate. In this script, we libraried in biomart. We also libraried in seq_nr. These two packages have at least two functions that have the same name. GetSequence, biomart has a getSequence function, and seq_nr has a getSequence function. And I have had issues if I were to copy and paste this whole thing. I have had issues where with biomart loaded, when I then try to load seq_nr and use it, there's issues. So my recommendation is if you're going to use seq_nr, open R fresh, don't library in biomart, only library in seq_nr. So I am not going to library in biomart right now. Get seq_nr in here. Let's tell seq_nr which repository we want to talk to. We're going to use GenBank. And then I'm going to scroll down to the section that talked about GC content, and let's go get sequence information for a gene. So we are going to get sequence information for the gene with HG and C symbol RPOB in 2 different bacterial species. So the first one is Mycobacterium tuberculosis. The other one is E. coli. Query is the main workhorse function for get or seq in R. So let's download the sequence for this gene from these 2 different species. Okay. Remember that what we just got out. So if you were to print this out, let's see what it looks like. First, let's ask what's the class. What kind of object is this? It's a QAW object, which means nothing to me. It's a specialized object, which means it's not super easy to just look at. If I just try to print it, I get cryptic description. I think sometimes an object will have sub-objects that are kind of included as part of it. And you can see whether those are available by doing names of that object. I'm going to do names, RPOBMT. And some of this we won't look at. The piece that I want is in the REQ element. What is the class of that element? I can access that element by using $. So tell me about this little sub-object called REQ. What kind of class is that? It is a list. What is the length of that list? 794. What does the first entry look like? If it's a list, how do I subset out the first one? I would use double square brackets. To pull out an element of a list, I use double square brackets. So the first entry is the first sequence that we got out for the RPOB gene in the tuberculosis bacteria. And we're told it has a name. The length of that sequence is 128, and then a couple other items that we won't worry about. So the actual sequence data is somewhere, it's described in this REQ element. There were 794 entries for tuberculosis. There were 263 for E. coli. Uh-huh. There, we have 2 objects now. We have this one, _MT for M. tuberculosis, and we have this one, _EC for E. coli. So, I don't think this has any name that's specific to the organism. I think this is just the name of the protein product. If I searched for that, I wonder what I get. Let's try GenBank code. So the code is apparently specific to our species. That name is specific to this gene in this species. So there would be a different name, or what it's calling an accession number, for the E. coli sequences. Okay, so there's lots of these sequences. And let's compare one of them. Let's find a couple of sequences for M. tuberculosis and E. coli to compare in terms of the proportions of the different letters and in terms of GC content. Let's see. The textbook referred to an entry in M. tuberculosis named that, and an entry in E. coli named that, because apparently those are pretty similar in function. So let's see if we can find them. Every year I use this code, these numbers change a little bit because the data from GenBank changes a little bit. Let's do JX303313. So that's not the right one anymore. Let me see if I can just go down a couple. 331. Oh, I want to go up. 3316. JX303316. That's the one I want. So I'm going to change this to 649. And then I think this one is still pointing in the right place. So I would like the name of this to be this AE05174 ... No, I want to do E. coli 1 005174.rpob. Okay, these are the two sequences that I want to play with. So let's get their actual sequence. I do get sequence, and I specify which entry of that list I want the sequence for. So for example, we now have this and that. They're kind of long. Let's tabulate the proportions of each of the letters in these two different sequences. I can use the table function. So table will take a vector of input, and it will tabulate how many times did each unique value show up. And I can just print these, TTMT, TT coli. We might change those to be proportions. Let's do TTMT divided by length, TTMT. No. What's that? That's what I'm trying to do. Yeah, you're right. RPOB MT seek. Thanks. And so those are the proportions. 19% of all the letters in M. tuberculosis are A's versus 25% A's for E. coli. Who has more GC content? Who's got the higher GC content? MT, because these numbers are bigger than those numbers. This Y out here, just FYI, if I remember correctly, that means there was some uncertainty about one of the bases, and I think it's only known to be either an A or a T, or it could be either a G or a C. They're uncertain about what that specific one was. I think there's only one out of all of them that was given that code Y. Okay, that's what I just did. GC content you could do easily from what we already have. So if I take the second and third entries of the table and just compute the proportion of that total. So sum up the second and third and divide by the total sequence length. And there we can see what we saw earlier by just looking at the table. There's more GC in M. tuberculosis than in E. coli, higher GC content. So suppose we had a sample from a community of bacteria living in some sludge on a lake or something. And we see evidence of multiple species in there. We don't know what there is there right now. We just have a spoonful of goo in which there's lots of bacteria. We're able to sequence that spoonful of goo and figure out that some of it is bacteria X, some of it is bacteria Y. It looks like there's two different species. What do we know about these species? One really high level way to distinguish them is GC content. One of them has really high GC content, one of them maybe has low GC content. So just right off the bat, you could say something like the first one is more amenable, more robust to things like extreme temperatures. All right. Suppose you had a protein sequence. So here's a list of amino acids for a protein, and you want to do something similar, like tabulate how many times do each of the amino acids show up in this sequence. I am making one up. This differs from this sequence. How? What's the difference between this sequence that I just created and this sequence? Okay, right. These are DNA. These are amino acids. How else do they differ? In terms of the way that R views this object, what's the length of my seq, do you think? It's just one. It's one character value. It could be hello or my name is Alan or something. All it is quotation mark stuff. The length of this object is 4,000 something because each of the letters have their own element. So if I wanted to tabulate, do a table, it would not work on this as it is. Let's try it. Table, my seq. All it's going to tell me is there's only one unique value. That unique value was that, and it happened once. So what I would like to do in order to tabulate is split those into a vector with an entry for M, then an entry for T, entry for M, ... So FYI, there's a handy function in R that does that for you. It's called string split or str split. So if I say string split my sequence, the one that we just defined, and let me do the help file for that, str split. So tell the function your vector that you want to split up, and the other piece that I specified was this argument. Notice that this argument doesn't have, this argument has fixed equal to false. If there's an equal to something, that means there's a default value, which means you don't have to use that argument if you don't want to. If I don't say anything about the fixed argument, R is going to use this value. Split does not have a default argument, so I have to provide something here. And it is described as a character vector containing regular expressions to use for splitting. That's not super helpful for us. It looks like I could say split equal to 0. I think I got split equal to quotation marks from an example. Basically, I want to split, I don't want to split wherever you see an E or something like that. I just want to split in between every single character. So by saying string split with like no space in this quotation mark gives me what I want. So my seq becomes that. And now we can tabulate. And we could compute proportions as well. All right. So let's think about if we have two or more sequences, how would we line them up? This is very relevant to bioinformatics today. If you do next generation sequencing and you get these reads, short sequence reads from a tissue sample, maybe they were obtained for mice. You're studying mice. So you do next generation sequencing and you get all these short little reads. What you want to do is map those back, map all those reads. Here's your pile of all these short little reads, sequences, and then you have the total genome for the mouse. I know that if you start at chromosome 1 and just start reading, this is what it is for a mouse. So here's one of my little reads, pick it up, short little sequence. What I need to do is find out where do I put that in the genome. Did this come from gene RPOB or did it come from gene whatever? So I need to align a sequence with another sequence. And obviously if we're talking about bioinformatics and next generation sequencing, it's a big task because we will have millions of these little reads. And each one of them needs to go back and find its place in the whole genome. So this idea of what's the best way to line two sequences up is important. All right. So let's go here and talk a little bit about what sequence alignment involves. So sequence alignment is arranging the sequences of DNA, RNA, or protein in an optimal way, as well as we can. Here's a picture that's worth looking at. Here is one protein, a subset of the protein, in the sequence for it in humans, mouse, rat, cow, and chimpanzee. And notice that the two, or the different sequences don't match perfectly. For example, right here in this location, human has an S, mouse has an A, AAS. There's differences. So in terms of aligning, if you required a perfect line up, you would never be able to do this. So it's a question of getting a, quote, good alignment in such a way that it minimizes mismatches and gaps and stuff. If you think about the evolution process of going from one organism through mutation and things to another organism, being able to, or differences can occur, like we're looking here. This is the same protein, does similar things for all these organisms, but it's made up slightly differently for the different organisms. And the way that can happen is through mutation over time, through gaps being created in the sequence, or through deletions happening in the sequence. But if you compare, say, humans to chimpanzees, and you see that there's relatively little difference in sequence composition, then you can say probably those two species are connected relatively recently by a common ancestor. Whereas if we looked at humans versus E. coli or something, there'd be very large differences in the sequences, and so we could say they probably diverged much longer ago. So sequence composition, aligning sequences has multiple uses. One is for figuring out lineages or probable lineages of how organisms developed over time in an evolutionary process. I just mentioned this buzzword. There's this idea of a conserved region of the DNA. And what that means is, like in this particular example, there's a lot of places in this particular protein where the sequence is exactly the same for these 5 species. We would say, or the biologists would say, these regions are conserved. They're highly conserved across this process that includes all those species. If something is imperative for survival, it's probably not going to be able to change much over time. Because if you get a mutation in a gene that controls something critical to life, you'll probably die, and you won't pass that on to your progeny. So conservation, conserved versus non-conserved. If you can find, say, a sequence that is conserved between 2 species, and suppose you know a lot about what that gene does in one of the species, but you don't know what it does in the other species. If you can do this alignment thing and find, oh, that's very similar to what we have in this other species, you can try to say, if it does this in the chimpanzee, it probably does something similar in the human. Okay, so there are 2 basic categories of alignment depending on the type of algorithm that you want to employ. There is a global alignment and there is a local alignment. I'll show you a picture in a minute, and I'll show you in R how to do both. Roughly speaking, global alignments take 2 sequences and they try to find the best way to line them up as a whole. So I'm going to take this sequence and that sequence, and I'm going to score matches and mismatches and everything across the entire spectrum of both sequences. Whereas a local alignment doesn't worry about whether it aligns well globally overall, as long as it finds a specific spot somewhere to align well. If you were to think about the situation where we have millions of short reads and we have the whole mouse genome, and we want to map all those reads back to the mouse genome, I would like to do a local alignment in that case because each little read is tiny compared to the huge reference genome. So I don't expect them to line up well globally, but I think there's going to be a spot in the whole genome where they line up well. So depending on what the application is, you might be wanting a global alignment or a local alignment. Both of these, in the way that we're going to see them, are just algorithms, and an algorithm is going to find the best solution it can given the parameters that you tell it. If there's a mismatch, let's do a penalty of -2. If you have to create a gap in order to put these 2 together, do a penalty of -4. If you have to extend the gap to be even longer, do an extra penalty of -1. Stuff like that. If you define those terms, then there are these algorithms that will go through and find optimal solutions. Okay. Here's a picture that kind of illustrates the difference between a global and a local alignment. So 2 sequences, 2 amino acid sequences that we're going to try to put together in an optimal way. If you use a global algorithm on these 2 sequences, you get this answer. If you use a local algorithm, you get that answer. The global task is trying to map the entire second sequence to the entire first sequence. So minimize penalties across the whole thing. The second one is okay with there being, say, these big gaps at the ends, as long as these local places where we actually line things up are good, are tight. These are the two main algorithms that you'll see referenced. The global alignment technique or algorithm is called Needleman-Wunsch. I guess that's how you say it. And the local alignment algorithm is called Smith-Waterman. We'll learn the Smith-Waterman algorithm, and you'll actually implement it manually. Another issue is whether or not we have just two sequences, or do we have more than two sequences that we want to put together simultaneously. If I'm doing my pile of reads and mapping them back to the mouse genome, I have two sequences at a time. I want to take this read and map it. So that's a pairwise alignment. Two things, just line those two things up. That's a relatively simple problem, relatively. All of these methods are computationally challenging, but say pairwise alignment is simpler than multiple sequence alignment, which is the idea that I now have, like in the picture from the very beginning, I have human, cow, and all that. I have 5 different species or 5 different sequences, and let's jointly align all of them in an optimal way. That's a harder problem, but there's algorithms for it. So for pairwise alignment, there are a few ways to do it. We're going to focus on that Smith-Waterman algorithm that I'll talk more about as soon as we have time. We'll also make one of these pictures. This is called a dot plot. So if you have two sequences, what you can do is make a picture where basically you create a grid where there's a separate row for each base in the first sequence, or a separate column for each base in the first sequence. And there's a separate row for each base in the second sequence. And then if I go to the first column, the first letter in the first sequence, let's say it's an A. If that matches with, okay, so here's an A in the first spot for the first sequence. Where are there A's in the second sequence? Anywhere there's an A, put a dot. So this like dot right there that I'm kind of hovering over would indicate that whatever that first base was in the first sequence, that same base showed up there. It also showed up there, and so on. So you get lots of dots all over the place. What you're most interested in is like stretches where there's like a continuous diagonal. Suppose that these two sequences were exactly the same. It's AA, TC, ATTG, whatever, and the exact same thing in the second sequence. Then for the first element, where does an A happen? It happens in the first element for this guy too. It'll probably show up in other places as well. So there will be dots for other places, but the most important one is this one maps to that one. And for the second one, there would be a dot on the diagonal, and the third one. So you would see a line, diagonal line, indicating good alignment between these two sequences. If you take two sequences and make a picture like this and it's just a big mess, they don't line up well. If you make a picture like this and there's little stretches of diagonals, that indicates that there are regions where they line up well. Or there might be a diagonal off here somewhere, a diagonal line off here, which means that there's a portion of this sequence that lines up well with that sequence, but they're shifted somewhat.
So in this case, rows would be specific?
Rows, there's one column for every entry in sequence 1, and there's one row for every entry in sequence 2. So there could be more rows than there are columns, for example.
Pairwise, not multiple.
This is pairwise. Scatter plot, two things being plotted against each other. We can make this in R. Yeah, I'll show you how. Couple more comments real quick on this article. Phylogenetic analysis, I was just going to mention. Has anybody ever done anything with phylogenetics? Yeah? Okay. So I'm not an expert in phylogenetics, but phylogenetics largely deals with the evolutionary trajectory of an organism going to other organisms. And a critical part of that, of creating that kind of tree structure, is sequence alignment. Here's 20 different species. Let's align them to each other, and the species that are really similar in sequence, we're going to put them close together in our tree. And then ones that are different and more different, they are further and further down the tree. So you can get pictures like this, which show common ancestor, and then we think there was a split into bacteria and archaea, eukaryota, and here was a split into this and into that, and so on. And the relative portions or the relative distances and stuff speak to sequence similarity or dissimilarity. And one other thing, just a couple of comments about other biological uses of sequence alignment. I'll skip that for now. Okay, so what I wanted to do next is go to some slides. So I posted under notes on eCampus a file called alignment.pdf. I've got it right here. These are some slides I found that I thought were pretty relatively readable. If you go and read up on Smith-Waterman, for example, or pairwise alignment, some of the documentation is not easy to understand. Like the first slides that I found were some slides from electrical engineering PhD program at MIT, and it was very computer sciencey beyond my abilities. So I like these slides. They show you how these algorithms actually work. So let's jump to the fun part. Let's go here. So this is slide 9. And let's figure out how do you do an optimal alignment of two simple sequences. These two, GAAGA, aligned with GTTTAAG. So we have to define what happens when there's a match, what happens when there's a mismatch, what happens if we have to create a gap in order to line these two things up. Let me just show you the finished product so you know where we're going. Here's the answer to this question. There are 6, there's actually, according to my count, 8, but these are 6 alignments that have the same score. These are all optimal alignments of those two sequences. So one way to optimally align would be to take the first one, which was just GAAGA, and stick it as is, lined up with the first G of the second sequence. If you do that, the first sequence was shorter than the second one by 2 bases, so we have to create a gap of length 2 in order to put those two together. So how did I do? I got a match, I got a mismatch, another mismatch, another mismatch. Here's a match, and here's a gap of length 2. If we use what we're going to define as the penalties and rewards for those different combinations, add them up, you get a score of -7. But there are different ways. I could do G gap A G A, or G A gap G gap, and so on. All of these result in a alignment score of -7. So here, how do we get these scores? Step 1 is to define the penalties and rewards. And there's no perfect answer. The slides use these. So if there is a match, reward yourself with a +1. If there's a mismatch, it's a penalty of -1. And we'll talk more about this soon. But a gap, I think every single gap, every single base that is treated as a gap gets a penalty of -3. So if I have to put in a gap, that's -3. If I have to put in a gap of length 2, that's -6. Gap of length 3 is -9, and so on. If I have to put in 2 gaps, one of length 1 and here's another one of length 1, that's going to be a penalty of -6 as well. So every one gap you have to do, we're going to penalize by -3. All right, so here's what you're going to get some practice doing. Take the 2 sequences and start by writing a table. So what you do is you put one of the sequences on the top and the other on the side. You put a gap or an empty cell here, and then you fill in with some numbers. And the way these numbers go in is if I were to create a gap in the first position. So instead of putting G with G, I'm going to put a gap and then move the G over. The penalty for creating a gap is -3. If I had to move it over 2 spaces, that's a gap of length 2. My penalty is -6, 3 spaces, 4 spaces, 5 spaces. Same thing going down. When I move horizontally, I'm creating a gap in the sequence that's at the top. When I move vertically, I'm creating a gap in the sequence on the side. If I move, okay, so that's there. In these cells, these -3s and the gap penalties are in that first row and first column. This row, this column. Inside, like in this cell, if I were to map G to G, I got a match. Those match. So my score for that particular assignment is a +1. If I were to map a G to an A, that's a mismatch, my score is -1. Here's a G with an A penalty. Here's a G with a G reward. That's step 1. Now we're going to fill in scores that accumulate as you consider possible alignments of one with respect to the other. Basically out of time. When we move, I said this before, when you move horizontal, so let me just show you this slide. I'm now going to put in some new numbers that I'm going to have to explain next time because we're out of time. But we're going to fill in this table. I'm going to fill in this table. We end up with something kind of messy looking where I have all these numbers in the bottom right of these cells. And then the question is, what path can I take in order to get from the top left down to the bottom right? For example, the blue arrows tell me, first put this G with that G. We went from there to there. Next, put this A with that T. We went from there to there. Then this A with that T. We went from there to there. Horizontal or vertical shifts are gaps. Diagonal moves are lining up with those two things. So this diagonal
arrow right here means that A is going to be put with that T. And this G is going to be put with that T. And then this A is going to be put with that A, and then gap, gap. And that results in a final score of -7. So this is the best alignment score we can get, according to this algorithm and according to our choices of rewards and penalties. So one of the optimal alignments is I can figure out by tracing this trajectory that goes from top left to bottom right. Here's another one, the green one, also ends up in the bottom right. So if you compare those arrows with this slide, you should be able to find those alignments. We'll practice that next time, and you'll get some practice doing it in the homework. So number 2, for example, you're going to do a global alignment of these two toy sequences. Here are the penalties and rewards to use. Write down the score matrix. That's what I'm referring to. I'm referring to one of these things. You don't have to do the arrows. It gets messy. But all the numbers. Put all the numbers in there. And then tell me what the optimal alignment or alignments are and what's the corresponding score. The good news is software is available to do this in a rapid way for us, but it's good to know kind of what it's doing in the background. Do it by hand. The R code will give you the score and the alignment, which will allow you to check your answer. All right, questions? And I will see you Friday. Thanks.